for you for the background noise, so just ignore. And moving towards the chapter number 5 of your biology, mitotic cell cycle. So, as you already know, that there is a video on live stream of mitotic cell cycle where I briefly describe about the stages of the mitosis, including some informations. So, if you haven't watched it yet, just go and watch it out and then start this video of the mitotic cell cycle. And for this, I think so that this PDF is also helpful for you. I have two PDFs, one on which I have already made the video, and this is another the where we are also understanding the structure of the chromosome, which is essential in this chapter. So what are the chromosomes? Basically, they are the thread like structures made of DNA. All right. And as you can see, there is a single chromosome. Where are the what is basically the chromatids is the one half of two identical copies of a replicated chromosome. It means that there is the chromatid is the one half of the chromosome. When two chromatids meet to the centromere, centromere is the point which holds up the two chromatids to form one chromosome. And this forms basically the duplicated chromosomes in metaphase, through which they just are basically identical, and the genes are being copied to each of the chromosome exactly to the each and the one, and then just separated to make two identical daughter cells. And this is the kinetochore. Kinetochore means basically this is the part where the centromere is attached to the microtubules during the mitosis. All right. So, what is a centromere? Is the region of a chromosome to which the microtubules of the spindle attach via the kinetochore during cell division. And what are the telomeres? And the telomeres are very essential. Is that protective sequence of nucleotides found at the ends of chromosomes. These are found at the end of chromosomes, as you can see in the figure. That telomeres are found at the end of the each chromosome. Alright, so DNA or deoxyribonucleic acid. DNA is the molecule of inheritance and is made up of series of genes. A gene is a section of DNA that codes for one protein. The gene consists of genetic modification, and the DNA molecules are in sister chromatids so that their genes are identical. Okay. When cells divide, one chromatid goes into one daughter cell and the other to the other daughter cell, making the daughter cells genetically identical. And the size of a DNA molecule is 2 nanometer. Alright, so this is the size of a DNA molecule. Okay, 2 nanometer, not of DNA. Alright, and then the total length of the DNA 46 chromosome of an adult human is about 1.8 meter. So 1.8 meter is essential for you to remember. All right. Uh, yes, exactly. The size of the DNA and the size of the chromosome is essential for you to learn. So 1.8 meter. This needs to be fit into a nucleus of 6 micrometer. So a 1.8 meter, how can be fit to the 6 micrometer? Therefore, the DNA is wound around the outside of proteins called histones. Histone proteins, basic proteins, which associate with DNA to form nucleosomes. These histone proteins and therefore nucleosomes condense DNA to make chromatin. Sorry, DNA to chromatin. Chromatin is a complex of nucleic acid, DNA, and RNA. RNA means ribonucleic acid, and other proteins whose primary function is to compress DNA into a compact unit which will fit into inside the nucleus. So, nucleosomes is not the part of your syllabus. The telomeres is the part of your syllabus. The telomeres are the ends of chromosomes and are needed for successful cell division, permit continued replication, prevent the loss of genes. It means in the diagram, it is very visible to you guys. Exactly. This is very visible to you guys as the telomeres and caps that protect the chromosome. It means that when the when the chromosome replicates and then replicates and then replicates, so by the replication, it loses the gene from the corner or from the side. all right, or from the ends, what you can say. The telomere is present at the end to just make sure that every gene is copied in the replication. All right. As cell divides over the time, telomere shorten and eventually cell division stops. All right. Telomeres are made of DNA with short base sequence that are repeated multiple times. The copying enzyme telomerase stops a little short of the end of DNA. So telomerase is the enzyme the telomeres in the chromosome which make sure that every of the gene has been copied. More bases are added which don't have useful information but allows telomerase to complete copying the meaningful DNA. And this is the cycle which I have already discussed. G1, S, and G2 phases, and then the cell growth, which is known as the mitosis, and then the prophase, metaphase, anaphase, telophase, and at the last cytokinesis, which means that the cell cytoplasm division, and after which it goes towards either G0 phase or G1 phase. So the interface is G1 plus S plus G2 and longest phase normal cell work functions the cell grows to its normal size time spent between cells division where the G1 cell make RNA enzyme and other proteins needed for growth. At the end of G1 phase the cell becomes committed to dividing or not. S phase is the synthesis of DNA where the DNA replication occurs. G2 phase is again the cell continues to grow, new DNA is checked, and any error are repaired. Alright, preparations are made to start cell division. A sharp increase in production of protein tubulin is observed, which is needed to make microtubules for mitotic spindle for the mitosis. Phase uh, nuclear division that produces two genetically identical daughter cells, maintains genetic stability, ensures cell retain function, maintains chromosome numbers. And these are the phases. The function of mitosis is very important as mitosis have the function of growth, all right, as it grows the cell and the cell replacement. Replacement means that when the cell dies, so it grows new cells and the cell replaces. Repair of tissues as sexual reproduction and immune response. In the sexual reproduction, you can take the example of amoeba, how the amoeba is sexually reproduced. And the immune response means cloning B and T lymphocytes. The stages of mitosis you can also learn is at PMAT. It means that prophase, metaphase, anaphase, and telophase. Where the prophase means that the centrosome migrates to opposite poles of mitotic spindle, chromatin starts to condense. Nuclear envelope and nucleolus begin to disappear. Centrosomes and migrate to opposite poles of the cell and forms poles of mitotic spindle. Esters and spindle fibers form. Metaphase. Centrosomes are at pole of the cell and organize the production of a spindle microtubule. Chromosomes line up across the equator of the spindle. Each chromosome is split at centromere. Chromosomes start to be pulled apart by microtubules. Anaphase. Chromatids move to opposite poles. Centromere are first pulled by microtubules. Spindle fibers contract, and chromosomes are pulled to the opposite ends. And at the telophase or the last stage, where the nuclear envelope and nucleolus reforming, the nucleus divides. The chromosomes are at the poles of the cell, and the diagram shows the cytokinesis, means the division of cytoplasm. So this is what basically we have learned in the previous live session also. So the cytokinesis means the division of cytoplasm. Cytokinesis in plant cell. Okay. Cell plate forms across equator of cell, cellulose, cell wall laid down. Cytoplasm is held divided into two. This is how the Golgi vesicles are also visible to you, and the cell plates are visible to you, and this how the cell plates are being made through which the cytoplasm divides. Cytokinesis in animal cell. Okay, contractile ring forms, pushing the equator of cell inwards, forming a cleavage furrow. Okay, this cleavage furrow deepens as active filaments in the ring contract. Okay, eventually the cell splits into two. This is very simple, as the visible terminologies have been shown in the diagram. Okay, differences in plant and animal cell mitosis. Plant cells don't have any visible centriole or ester. In plant cells, dotted separated by cell plates, there is no cleave furrow formation. Okay, centromeres, centrosome, and centrioles. This is maybe sometime very complicated for the candidates because the centrosome and centromeres and the centrioles are having the a bit type of different meaning, but the words are being sometimes pronouncing as same. As I'm always confused in the centrosome and the centromeres. That's for me. So in the centromere, this is needed for separation of chromatids during mitosis. Centromere is present at the where is centromere present in the chromosome. Site of attachment of spindle nucleus. Sorry, spindle microtubule. It means that it is attached to the spindle. Each metaphase chromosome has two kinetochores at its centromere and microtubule. What are the kinetochore? Protein molecules which bind to DNA into centromere and microtubules. Bundles of microtubules called spindle fibers extend from kinetochores to poles of spindle during the mitosis. Alright, and this is the visible diagram where the kinetochores are being visible to you. The kinetochores are at the side of the centromere and the spindle fibers. What is a centrosome? Centrosome is an organelle found in animal cells that acts as the microtubule organizing center, or MTOC, for construction of the spindle, present at the poles of cell. And the centrioles act as MTOCS. It means that this is um, microtubule organizing center. Assemble or forms spindle fibers during prophase. Contraction of spindle fibers during anaphase separates sister chromatids. What are the stem cells? This is very important. Okay, this I does not have explained in the previous live session. Okay, so what are the stem cells? So first of all, you think that maybe this is present in the plant. So no, it is present in human being. Stem cells are cells that can divide an unlimited number of times by mitosis. As you can see that the one stem cell is dividing into three more stem cells. And furthermore, to make the specialized cell. Extent of the power of a stem cell to produce different cell types is called its potency. Stem cell therapy, introduction of new adult stem cells into damaged tissues to treat a disease or injury. Okay. Cancer. Now, what is a cancer? A result of uncontrolled mitosis or cancerous cells divide repeatedly and form a tumor. Carcinogen. What is carcinogen? Agent that causes cancer, such as radiations. It might be UV, means ultraviolet, or X-rays. Tar or nuclear fallout. Cellular changes that occur in development of cancer. So these are the stages. Mutation occurs in gene responsible for cell division, leading to formation of an oncogene from a proto-oncogene. Okay, this causes uncontrolled cell division. Coordination of cell cycle is lost. Cell does not receive response to signals that tell it to stop dividing. It means that this is very dangerous. Loss of function and lack of specialization occurs. A tumor is formed, an irregular mass of cells showing an abnormal change in mass. Now just see the stages of the how the cancer being formed. So what is oncogenes? The mutated genes that cause cancer. Mutation means the unequal number of chromosomes or the change in the number of chromosomes arise due to carcinogens. Means the carcinogens or the oncogenes are the same which are producing the cancer cells. Example of carcinogens: any agent that causes cancer is called a carcinogen and is described as carcinogenic. The examples are ultraviolet light, tar in tobacco smoke, or asbestos and X-rays. Okay. In the second, the cancerous cell does not respond to signals from other cells, so continue to divide. Means it's dividing uncontrolled, with an uncontrolled number, and they have been dividing with an unequal number. Okay. At the third, the mitosis occurs, and at the fourth, the cancerous cells not removed by immune system. Now means it is enough stronger that it is not removed by the immune system, and they absorb the nutrients. After absorption of the nutrients, fifth is a rapid mitosis, and now you can observe the number of the cancer cells, and this how the tumor gets bigger. All right. So do you know what is a tumor? A large ball which consists of a lot of oncogenes. And the seventh, the tumor supplied with blood and lymph vessels. If it is a malignant tumor, tumor cells spread in blood and lymph to other parts of the body. If it is in malignant tumor, it is very dangerous and cannot be converted towards the undangerous in the condition, as it means that when it enters first or it becomes first a malignant tumor, it means that now it is enough dangerous. Metastasis. Metastasis is something very important. As the tumor cells invade other tissues, secondary cells form throughout the body. It means that now it means that the cancer cells have been arriving towards the blood vessel and the lymph vessels, and through which they have been around towards all organs or the particular organ of the body. The cancer how the at the stage of the metastasis, when the cancer completely harms or attacks and just have to invade the tissues or the organ, when it completely goes and just damage the or start the damaging of the uh, in a particular organ, this stage is called the metastasis, and at the metastasis a person get to know about he is having a at this stage. When he knows that he is having the cancer, there is no solution, because of the reason that now, because obviously he is knowing at the stage when his body is going to be destroyed completely, or the organ is completely trying to be destroyed, such as the lungs cancer, the kidney cancer, the liver cancer, or whatever the cancer cells are going to attack on that organ. Okay. So this how they attack, and at the last, the metastasis means now they finally attack and just invade, which is obviously then dangerous. And at this stage, the person got to know about his cancer. But at this stage, he can do nothing, and there's no better treatment till now being formed or being given towards us, and for which that's the last stage. And the metastasis is the stage after which a person confirmedly died. Means this is the last stage of the cancer, and for a person too, obviously, because after metastasis a person will be die. So that's what all. And I hope so that you understand about the telomeres and the structure of chromosomes, and also the cancer and the stem cells. So I hope so that this is very best video. If it does not understand the stages known as prophase, anaphase, metaphase, and the telophase, go through the first live session of mitotic cell cycle, where I have given a detailed account on it. Okay, so then till here, take care. And if it does not understand anything, you are allowed and feel comfortable to just comment below and wait for the reply. Till that, take care.